Today we're going to be looking at Unit 6. This is a long one, so it's going to have to be split into two parts. So this first part is focusing on gene expression, looking at things like transcription, translation, whereas the second part is looking at overall regulation. It was known that traits were inherited, but it was not really known how that process happened. The discovery of DNA's role in inheritance is arguably the most significant contribution to our understanding of how life works, but it wasn't the result of any one person, but the final result of decades of investigation. Friedrich Miescher isolated nuclein and its associated proteins, so he was the first to identify DNA as a distinct molecule. Thomas Hunt Morgan studied fruit flies, and he was the first to realize that the chromosome, the constituent components being DNA and proteins, were the prime candidate for genetic material because he figured out that there were traits, genes, that were associated with those chromosomes, specifically sex chromosomes. Levine was an American biochemist who studied the structure and function of nucleic acids, and he was the first person to characterize the different forms of nucleic acid, DNA and RNA, and components of the nucleotide, so he figured out what the nucleotide was made of. Griffith was the first person to figure out transformation, the idea that bacteria can uptake genetic information from their environment. That was further studied by Avery, McCarty, and MacLeod, who then realized that it wasn't just all components that allowed transformation to occur, but DNA. So DNA is the transforming factor. Building on the work of Levine, Chargaff determined that the bases of DNA have a characteristic relationship. At this point, it was known that chromosomes are made of two ingredients, DNA and protein, but we didn't really know which one was more important or which one was involved. But Chargaff realized when you compare the compositions of adenine and guanine and cytosine and thymine, that there was a characteristic relationship between A to T and G to C. There are exceptions to this rule, but we eventually realized that that was dealing with things like single-stranded DNA and other exceptions. So for double-stranded DNA, the typical DNA, Chargaff's rules apply. However, it wasn't until 1952, when Hershey and Chase studied the T2 bacteriophage, that we realized that DNA was the transforming factor and that DNA is the carrier of genetic information. They used radioactive phosphorus-32 and sulfur-35 to label DNA and proteins respectively. They were able to determine that the viruses, the bacteriophage, passed the genetic information of DNA into the bacteria, not proteins. So this radioactivity determined that DNA is the carrier of genetic information. Watson and Crick's main work was using the information from Rosalind Franklin's lab. They determined the double helical structure of DNA. So it, in their words, has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So building on the information of all that came before them and the work of Rosalind Franklin, they were able to fully figure out the double helical structure and then to propose at least an idea for how replication occurred. The two DNA strands, or polynucleotides, are composed of simpler monomeric units called nucleotides. Each nucleotide is composed of one of four nitrogen-containing nucleobases, a sugar called deoxyribose, and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds called phosphodiester bonds formed by dehydration synthesis. The nitrogenous bases of the two separated strands are bound together according to base pairing rules of A to T and C to G with hydrogen bonds to make double-stranded DNA. The complementary nitrogen bases are divided into pyrimidines and purines, with A binding to T with 2 hydrogen bonds and G binding to C with 3. DNA is typically a double-stranded helix with the two strands connected by those hydrogen bonds, consistent with Chargaff's rules. The DNA double helix is anti-parallel, which means that the 5' end of one strand is paired with the 3' end of its complementary strand, and that allows for the structure that we're used to thinking of as the double helix. The replication method proposed by Watson and Crick is what we would now call the semi-conservative model, with the two strands separating along the weak hydrogen bonds in the middle. In 1958, Meselson and Stahl wanted to determine how bacteria replicated with proof positive from experimental evidence. So bacteria were cultured in mediums containing a heavy isotope of nitrogen, nitrogen-15, and then transferred to nitrogen-14 for later replications. Based upon the samples, they were able to eliminate the first replication for the conservative model and the second replication for the dispersive model. So showing that the semi-conservative model was the most likely model for DNA replication. The process of DNA replication ensures that every new cell that results from mitotic division has identical DNA, and enzymes facilitate this replication process. So the two strands of DNA, when it's split, they are very, very different in how they work, but if you have an understanding of how the enzymes facilitate the replication process, you'll be most of the way there. Bacterial chromosomes have one origin of replication. The bubble forms at the ori, the initiator protein is binding, and it slightly unwinds the DNA using ATP. The replicon or replication bubble forms, and that circular chromosome will then be reproduced. But in eukaryotes, there are multiple replicons. There are multiple replication bubbles that allow this process to occur within the linear chromosomes. A large team of enzymes coordinate this process, but it all starts off with an initiator protein. Helicase simulates the separation of the two strands, unwinding the DNA at the origin of replication, which is an area rich in A to T bonds because it's weaker due to two hydrogen bonds. Topoisomerase breaks and untwists and rejoins the strands, relieving the strain caused by helicase and aiding in the unwinding process. The single-stranded binding proteins bind to the parent strands to prevent reattachment and stabilize the single-stranded structure. RNA primase creates RNA primers to act as a starting place for DNA synthesis. So all of this is going to work together to allow nucleotides to be added to the process. The polymerase breaks off the phosphate bonds of the nucleotide, allowing energy to be available for the binding. The two phosphates are released as a pyrophosphate, which can be recycled, and the new bond attaches. So the DNA always elongates from 5 to 3 because the energy is on the 5 and the open OH group is on the 3. The leading strand is the easier replication process to understand because replication is proceeding in the correct direction, for lack of a better word. Replication is proceeding from 5 to 3. So DNA polymerase 3 is our main DNA builder. It's able to synthesize DNA onto an RNA primer. Why do we have to have the primer? Well, DNA polymerase can only work on a double-stranded structure. So if there's only one strand, DNA polymerase cannot do anything at all. But because the replication is proceeding in the 5 to 3 direction, it's going to be very easy. The DNA structure is going to be continuous, and that's really it. So the leading strand is pretty straightforward. The lagging strand is a little bit more difficult. Because replication is trying to occur in the opposite direction, we have to work against the flow of the enzyme. So the lagging strand, the RNA polymerase, or the RNA primase, it continuously creates new primers for DNA polymerase 3. This creates a discontinuous strand, which those fragments are called Okazaki fragments. What's really happening is the DNA is looping around so that the direction is artificially in the correct direction, allowing the enzymes to flow. It's still DNA polymerase 3, but it works with multiple primers and produces Okazaki fragments. Once all of the complementary bases have been produced, we need to enter the process of termination. In termination, you're going to have a variety of enzymes that come together to clean up and to finish off the process. DNA polymerase 1 removes the RNA primers and replaces them with DNA. Polymerase 1 is a slower builder, but it does allow the double checking of polymerase 3 work, proofreading the DNA strands and correcting any errors. DNA ligase will seal the Okazaki fragments together, connecting the leading and the lagging strands by forming phosphodiester bonds. In addition, ligase will work with nucleases and polymerase to correct any errors in DNA sequencing. It's not 100% accurate, but it does reduce the error rate significantly. Our only real problem now is the final primer of the lagging end. Because DNA polymerase can only work on a double-stranded structure, it can't move into that 5' polymerase or 5' primer place to replace it. So that means that DNA is going to be shorter every time that it replicates, and we're going to have to find ways to deal with that. So as previously mentioned, the linear chromosomes face an issue not seen in the circular DNA replication. So due to the fact that the RNA primer is required for initiation, the lagging strand is going to be shorter. We find at the end of DNA we have telomeres. Telomeres are expendable non-coding sequences at the end of DNA. For vertebrates, this is AGG, GTT, with the complementary strand being TCC-CAA. The sequence is repeated thousands of times and is there to be lost. So eventually the telomere is lost And when you start losing useful DNA, that's what's going to lead to cellular aging. Now, cancer cells and other organisms have found ways to fix this problem. Telomerase, for example, is an enzyme that allows the repair of the ends, or you could loop the DNA into itself to create a temporarily triple-stranded structure. So mammalian telomeres, there are ways to fix it, but we don't really have good solutions, as typically it's cancer that's doing this. So telomeres are shortened as we age, but telomeres can also be shortened by things like stress and smoking and obesity and lack of exercise and a poor diet. So telomeres are something that we're really looking into as a broader problem, not just as a DNA replication one. We do see that the actual enzymes that we're looking at are all tied together into a broader structure called the replosome. So for prokaryotes, for example, each dividing nucleoid requires 2 replisomes for bidirectional replication, and this continued replication at both forks in the middle of the cell, and then you reach the termination site where it is released. For eukaryotes, there's numerous replication bubbles forming at origins of replication throughout the chromosome. As with prokaryotes, 2 replisomes are required, one at each fork, located at the terminus of the replication bubble. Because of significant differences in chromosome size and complexity, various aspects of eukaryotic replication, including terminal phases, are really less well characterized than for prokaryotes. So there are some fundamental differences. But overall, we find that the enzymes are connected together, allowing them to work at the same speed, at the same pace, and maximizing the efficiency of this process. The functional segments of DNA which code for the transfer of genetic information are called genes, and the genetic code is a universal across all domains of life with a few exceptions. This triplet code, which reads in units of 3 bases, called a codon, codes for all of the possible combinations of amino acid. These combinations are used to specify the 20 different amino acids used by living organisms. Well, actually 22, but one's rare and one only occurs in archaean. But this code is redundant and unambiguous, allowing for an increased efficiency of transfer. But that universality of the code shows us that this is something that comes from common ancestry, or LUCA, rather than any other process. When you actually look at gene expression or transcription and translation, we find that there are some fundamental differences, the most important one being that eukaryotes have a nucleus, and that drastically changes this process, because DNA molecules do not leave the nucleus. Protein synthesis must occur on the ribosomes in the cytoplasm, so the codes must be carried from the nucleus to the cytoplasm. For a bacteria, that's not a problem. There is no separate component here. So for us, we have multiple steps. So the process of gene expression in bacteria is significantly simpler and significantly faster. For us, that complexity allows for a greater degree of control, which is important. We don't have a ton of enzymes to memorize for this process. You really only have one, or at least one category. RNA polymerase is going to bind to a promoter sequence on DNA upstream or in front of the targeted gene and begin the production of mRNA. If you're a prokaryote, that enzyme will bind directly to the promoter as shown here, and that process will occur. If you are eukaryotic, the enzyme is going to require transcription factors to bind, and that's where that Tata box promoter is going to come in. Which RNA polymerase is used in eukaryotes depends upon what type of RNA is being transcribed, and we'll look more at that in a moment. So eukaryotic transcription, you're going to have the Tata box promoter that's going to aid in binding. You're going to have general transcription factors that aid in the process of RNA polymerase binding, and then you're going to have specialized ones that allow for specific mRNA upcycling, and at least upregulating. That process creates a binding site that allows RNA polymerase to connect and allows the production of mRNA that will proceed in the 5 to 3 building, so 3 to 5 reading. The template strand is the strand of DNA that the transcript is being produced from, and the coding strand is the complementary one. RNA polymerase unwinds DNA bases, about 20 bases at a time, and reads 3 to 5 and 5 to 3 like we said before. There is no proofreading to this process, so errors can occur, but the general process is going to be consistent. In bacteria, transcription proceeds through a termination sequence in DNA, causing polymerase to detach. In eukaryotes, RNA polymerase II, which is the RNA polymerase that builds mRNA, transcribes a DNA sequence called the polyadenylation signal, causing proteins in the nucleus to bind, and then about 10 to 35 nucleotides downstream, those proteins will cut the transcript free, releasing the pre-mRNA. In eukaryotes, mRNA is processed after transcription and before it leaves the nucleus through nuclear pores. The 5' GTP cap is added, which protects the mRNA from RNase enzymes and provides binding energy. The poly A tail is added after the polyadenylation signal, and it's somewhere between 40, 50, and 250 A's or adenines that protects the mRNA and helps it to leave the nucleus. And then we look at the insides. In the inside, you have introns, which are non-coding regions. These must be removed, and the exons, the coding regions, must be spliced together. This is done using small nuclear RNA in a broader protein structure called a spliceosome, pulling those pieces together and putting the exons. The introns are recycled, and the exons are going to be expressed, or the exons are going to exit the nucleus. There are currently more than 30 described types of RNA, but overall there are 3 common major types that you need to be familiar with. The mRNA, or the messenger RNA, carries DNA sequences to ribosomes. tRNA, or transfer RNA, carries the specific amino acids to the ribosomes for protein synthesis. And rRNA, or ribosomal RNA, is the major structural building block of the ribosome. Other ones we could run into: small nuclear RNA, or snRNA, miRNA, siRNA. So a wide variety of RNAs exist that have different roles, and each one is produced by different polymerases and different processes of production using the same basic techniques. Ribosomes are the structures where polypeptides or proteins are built. They're made up of protein and RNA, rRNA, and each ribosome has 2 subunits, a large one and a small one, which come together around the mRNA, kind of like 2 halves of a hamburger bun coming together around the patty. The ribosome provides a set of handy slots where tRNA can find their matching codons on the mRNA template and deliver their amino acids. These slots are called the A site, or the attachment site, the P site, or the peptidyl site, and the E site, or the exit site. Not only that, but the ribosome also acts as an enzyme, catalyzing the chemical reaction that links amino acids together to make the polypeptide chain. All of these things work together to allow this production. The tRNA specifically is carrying the amino acids, and the tRNA is just transferring these bonds around. There are enzymes that are specific to very particular anticodons that actually use ATP to attach that and then transfer it to the ribosomal structure. In translation, the anticodon of the tRNA with its attached amino acid pairs to the codon of the mRNA, which is attached to a ribosome. Specifically, the AUG UAC methionine carrying tRNA is going to come in and start this process. When a second tRNA with its specific amino acid pairs to the next codon in the sequence, the attached amino acid breaks from the first tRNA and is bonded to the amino acid of the second. The ribosome forms a peptide bond between the amino acid, acting as a catalyst, and the amino acid chain begins to form. The empty tRNA is going to move off and pick up another matching amino acid from the cytoplasm in the cell, and this sequence will be repeated until a stop codon is reached. So initiation is focused on methionine's binding directly to the mRNA of the small subunit, and then the large subunit forms around or binds around that existing tRNA, so that the methionine carrying is already in the P site of the ribosome. That moves along, and this binding of the second, the energy is used to transfer that bond, and that process occurs again and again and again, forming peptidyl bonds and readying the ribosome each time for the next amino acid tRNA or charged tRNA. Termination is going to happen when a termination signal is reached, a stop codon. Basically when the finished polypeptide chain can be released. The stop codon is going to trigger a series of events. A release factor is a protein that allows for the termination of translation by recognizing the termination codon or the stop codon. They're named this release factor because they release the new polypeptides. After termination, which is going to involve the binding of water to break that connection, so hydrolysis is going to occur. The polypeptide may still need to fold in the right 3D shape, but it will be released from this process. So overall, we see this general process with RNA polymerase creating the pre-mRNA. That pre-mRNA is then modified with the cap, the tail, and the splicing of the exons. That then binds to the small subunit of the ribosome. The tRNA binds as well, the large subunit connects, and that process continues until a termination sequence is reached. If this were to be considered a bacteria, obviously the nucleus would be removed and the mRNA would be directly used by the ribosomes rather than waiting. So that separation allows for a greater degree of control that doesn't exist in bacteria. So when you actually look at the process, we find that bacteria significantly outpace eukarya in this, because as soon as the mRNA is being released from the chromosome, as soon as it's coming from the RNA polymerase, the ribosomes can begin to attach and produce. So transcription and translation are not separated in space or time in a bacteria. But in a eukaryotic cell, the binding of ribosomes cannot happen until it's in the nucleus. You can still have multiple ribosomes attached, as shown here, the polyribosome, but there is a separation of time and place. Termination do not happen at the same time or place in eukaryotes, but they do in prokaryotes. An additional complexity to this process that happens in eukaryotes is the targeting of those polypeptides. Almost all proteins that are destined for the secretory pathway or to be released have a sequence at the N-terminus, commonly referred to as the signal peptide. That signal peptide is recognized by the signal recognition protein, and that determines whether the mRNA that is being produced by a bound or a free ribosome. So you actually have these little markers that are removed by the ER that tells the ER where to send the proteins that it is producing. So when you have multicellular organisms, we have to target these processes, and this is how we do that.